Okay, hi everyone. So today we're going to explore how T cells are activated in our immune system. Up until now, we've spent a lot of time talking about how T cells are formed. Remember, they are born in primary lymphoid tissues, like our bone marrow, but they actually need to go and go through an extra sort of finishing school, if you will, known as the thymus. Right, so this is where the uncommitted precursor cells that came from the bone marrow can be encouraged to become T cells. So that's where they undergo the process of VDJ recombination to make their own T cell receptor. And then through the rather tricky process of your tissues, your thymus, selecting for T cells that have T cell receptor that can properly associate with the HLA proteins that make up the MHC platforms on the surfaces of our cells, because that's very important, because ultimately if the T cell is going to go to work, that's how it's going to interact with the cells of your body, through binding to the MHC proteins that present different small little peptides to the T cell to see if any of those small little peptides are something that the particular T cell recognizes and your immune system should care about. Right. So we said that there are positive selection processes to make sure that the developing thymocytes can bind MHC, but at the same time, it's kind of tricky because if the shape of the T cell receptor that this thymocyte has made binds too strongly to MHC, then that's not good, right? Because then it's almost like if that T cell is allowed to go to work in your body, then any cell it bumps into that has MHC on its surface, regardless of whatever the little peptide is that the MHC is trying to present, if the T cell receptor binds too tightly to the MHC, then that T cell might just end up reacting against anything, right? Any of your body's healthy cells, and that would be really bad. Also, we learned that as the thymocyte is developing and makes a T cell receptor, it has to make both CD4 and CD8 co-receptor. And ultimately, depending upon which one of those have a chance to interact with the MHC that a brand new thymocyte is being trained against, that will determine whether that particular T cell ends up being a single positive CD4 helper T cell or a single positive CD8 positive killer T cell, cytotoxic T cell. And then to finish things off, right, great. You have these thymocytes that have successfully made T cell receptor. The T cell receptor works just right to interact with MHC, but not too tightly. Then you have to go through the whole process of allowing it to sample different kinds of peptides that it would encounter from all over the body to make sure that these T cells don't autoreact, right? And so we finished last lecture discussing how the epithelial cells of the thymus have a very special transcription factor known as AIR, right? Which goes around the genome and kind of randomly turns on little proteins so that these epithelial thymic cells, right, are making small samples of all kinds of proteins from around the body. And then those samples are chopped up into small peptides, loaded on MHC. And so the developing T cells have a chance to make sure that they don't auto-react against any of these. Only once that is done does the T cell have a chance to enter circulation and possibly go to work. Right? So let's take a closer look at how these T cells, once they're in your system and going around, how do they activate? Right? How do they actually properly know when they should get excited about something that they see, right? And then what happens after that? So this is a nice reminder. I always want to encourage you to think about, how B cells and T cells are distributed in your body, because that's really important in understanding how they do their job. So again, we said that both B cells and T cells are born in primary lymphoid tissues, such as red bone marrow. Right? So these are where they're really originally born. B cells, of course, leave the bone marrow already having made their immunoglobulins on their surface, and they have to be screened central tolerance training in the bone marrow to see if that B cell's brand new IgM causes any trouble, right? And if that's okay, the B cell is allowed to then go into the blood, where it undergoes peripheral training to make sure its immunoglobulin doesn't get into trouble binding, numerous totally normal antigenic signals that your body's healthy tissues are constantly making and providing, right? And if that B cell makes its way into a secondary lymphoid tissue, such as a lymph node or other examples of secondary lymphoid tissues like the spleen, for example, right? Then that B cell fully matures and then is allowed to go to work. And then for the rest of its life, it is recirculating back and forth through the lymph system, back into the blood, and eventually to another lymph node somewhere, right through the lymph system, back to the blood over and over. And as we've kind of learned to appreciate, the overwhelming chances are a particular B cell will never end up being useful to bind anything, right? At the same time, the T cell that leaves the bone marrow, this blue cell here, remember, when it leaves the bone marrow, it doesn't have anything resembling a T cell receptor on its surface yet, because it's still uncommitted. It doesn't know what it wants to be, right? So some of these, when it's released into the blood, will float around, and some of them will, by chance, randomly end up flowing through the blood, through the thymus, where they then exit and start hanging out in the thymus. And that's where they will then, through different signaling of thymic epithelial cells, be encouraged to become a thymocyte. They will try to do their VDJ recombination, make a T cell receptor. They will become first double positive for CD4 or CD8, right? If they are, in fact, an alpha beta T cell receptor. Remember, some of the thymocytes actually become gamma delta T cells, in which case that's the end of their sort of time in the thymus. They would just go into the blood and go to work. But the alpha beta T cells, the ones that we get more excited about, because those are the cells of your adaptive immune system that everybody talks about, your T cells, they have to then go through that whole positive and negative training and screening to make sure, right, that they know what type of T cell they are, CD4 or CD8, right? And they don't cause trouble. They don't auto-react to different types of self-peptides. So then they go into the blood, and it's the same story. They go through the blood, they take turns going into lymph nodes, going through the lymph system, right? And then back into the blood over and over and over again. And again, think about it, right? The overwhelming chances are a given T cell that you are following through this whole process won't ever amount to anything in terms of binding anything, right? But that's how our bodies do it, because we want to make sure, because we cannot risk the fact that if we are infected by some sort of a pathogen like bad clown, right, that our innate immune system can't handle, we better make sure that we have some B cells in our system, some T cells in our system that do recognize the antigenic signals from bad clown, so that we have a chance to mount a strong adaptive immune response to save our lives. All right, so this idea of passing between the lymph system and the blood, lymph system and blood, I think it kind of begs the question, right? Why? Why do lymphocytes like T cells and B cells need to go through the lymph system to places like lymph nodes in order to really do their work, right? Why wouldn't it be enough just to have them floating around in our blood, and wherever they bump into things that their antigen binding domains on their immunoglobulins or T cell receptors recognize, why can't they just do their job there, right? So let's talk about this, because this has to do with how evolutionarily we've organized our immune system, right? With these lymphocytes going between our blood circulation and our lymph. Here's one way to think about it that I think, is pretty straightforward. In your blood and in normal tissues that your blood is flowing past or flowing through, there are many more normal cells of your body that lymphocytes like B cells or T cells could bump into and will bump into, compared to pathogenic cells, right? I mean, I think intuitively, right, nobody can deny that that makes perfect sense. So if you think of it like these little lymphocytes going through the blood, so here you have some lymphocytes going through the blood, right? It's like a crazy place where stuff is just all over, right? All kinds of signals, and most of these are just normal signals. It's like as though you were a shopper wandering through a humongous flea market, right? Like the big flea market at Aloha Stadium back before the whole COVID thing happened, right? There's all kinds of fun stuff there, all kinds of neat junk and stuff like that. So imagine it's like a lone shopper wandering through a flea market, right? If you were really looking for something specific, in other words, there's something specific that you're interested in, right? You might say, yeah, you don't really go to the flea market expecting or knowing where to find that. You just kind of go and see what you see, right? So imagine if that is like an individual lymphocyte, like a T cell or a B cell, just flowing through your blood, right? Bumping into all kinds of things, even though there's really only one specific thing that you're interested in, right? For the T cell, that would be a particular peptide shape that's presented on the surface of the MHC platforms that the other cells that it's bumping into is showing it, right? For the B cell, it might just be any kind of shape, any kind of molecule, just as long as it presents this particular shape that your immunoglobulin looks like. So you can imagine that's not very efficient, right? What are the chances that a B cell or a T cell would really encounter, right, the particular antigen that it's interested in? So a much more effective way of encouraging lymphocytes to encounter the things that your body wants them to encounter would be to organize antigens in specific places, places that lymphocytes would visit to be able to have a much easier time checking out what's going on, right? So it's like shops at the mall, right? Like Ala Moana, for example, right? So our lymph nodes are like the stores organized in a big mall. These are specific places where antigens are concentrated. They are brought there so that when you have lymphocytes visiting the lymph nodes, it's just like a shopper at Ala Moana, going from store to store, seeing if there is what it's interested in, to potentially purchase. A lot of this idea of how antigens get concentrated into places like lymph nodes and how, when lymphocytes come to these lymph nodes, they are presented these antigens is very much like this example of a store and how products need to be delivered to the store and then advertised, right, or displayed for shoppers like yourself wandering through the mall to actually have a chance to see it. So this is the function of dendritic cells. We already learned that dendritic cells, are these sort of lovable immune cells that kind of like don't really have much ability to attack pathogens or other cells or whatever, right? So their job is just simply what? To go around collecting stuff so that it can present this stuff to other lymphocytes, other immune cells that might be interested in it. So let's imagine a scenario. Oh boy, one day, you're running around barefoot, it's Hawaii, right? So we always do that. But in this case, you stepped on a tack, right? And this is pretty serious, because look, you fainted, right? That's why you're lying here. All right, well, let's think what's going on at the site of infection, right? The moment the tack penetrates or breaches your tough outer skin layer, it carries with it bacteria, and the bacteria that was on the bottom of your foot also goes in, right? So that's the moment infection starts. So let's see what's going on here. So here we have the wound. Your tough epidermal layer is breached. So now you have bacteria or some kind of antigen, some kind of pathogen, right? It might actually be like a virus or something, gets into your dermal layer. And here you would have resident immune cells like macrophages wandering around and like dendritic cells wandering around in here. And so these dendritic cells would pick up. They would be phagocytosing these different bits and pieces of things that are in the tissue. And so that would include these red dots, which are maybe proteins or pieces of protein or other molecules that come from the bacteria. So the dendritic cells are going to have to package up, right, these red dots and transport them, just like packing them up in these boxes and loading them up in this delivery truck, right? And then the dendritic cell then, just like other cell debris or cells in the area, remember, in this tissue, there'd be capillary beds of your blood supply coming in, and draped over the capillary beds, you would have all these lymphatic vessels, right, that are busy draining extra stuff from the site of the tissue, like fluid, like waste products, and like things like pathogens and other cells, like just like the gutter at the side of the road, right? So the dendritic cell then will drain into one of these lymphatic vessels and be carried to the nearest lymph node. So let's pretend, for argument's sake, that this is the closest lymph node to where the site of infection is, right? I mean, the way they draw it on the diagram is a little oversimplified, right? Because I'm pretty sure that you have at least a couple lymph nodes closer to your foot than up at your knee. But they wanted to emphasize the fact that there is this necessity to transport things from the site of infection to places of secondary lymphoid tissues, like this lymph node, right? And this lymph node then is like the store at the mall. So then the dendritic cells then have brought stuff there and can now show, right, this antigen to lymphocytes and other immune cells that have come by the store to browse and maybe buy stuff. Okay? So this already suggests to you that a dendritic cell, right, isn't just this kind of simple little cell that doesn't deserve much credit, right? We sort of jokingly say it goes around just scrubbing up everything. It actually has to do a number of different tasks. It has to collect antigen, it has to transport antigen, and it has to present antigen. Right. So as a dendritic cell is doing these steps, it actually changes quite a bit in terms of morphology, so the shape of the cell, and in terms of what's going on inside of the cell that reflect these different stages of its activity. Let's start with dendritic cells that are close to the site of infection. So at the site of a skin wound, right, the dendritic cells are busy phagocytosing, or taking in pathogen and trying to process the proteins into small peptides, that then those small peptides can be loaded onto MHC. So in this figure here, under the microscope, we are looking at a number of dendritic cells, and they've used a couple of fluorescent antibodies to stain or indicate the presence of particular features and proteins. So the red stain that stains would stain different components of the lysosome, right? So that's the vesicle after bits and pieces of pathogen are taken in, right? The vesicle, the endosome, fuses with the lysosome that contains all these nasty sort of proteolytic enzymes, right? And then that becomes the phagolysosome, where proteins and things are broken down. Remember, that's also where MHC2 would be present to be loaded up with small fragments. So in this figure, green stain is staining the MHC proteins, so you could see where they are. Now you say, well, I kind of see some green. I don't see any red. I see a lot of yellow. Yellow is where the red stain and the green stain perfectly overlap. So what does that tell you? That means in this particular picture here, the majority of MHC proteins, the MHC platforms, right, that are being loaded up by being fused to the lysosomes, right? So that's where you really see that the red dots are being boxed up, ready for transport. Here you see the actual shape of a typical dendritic cell during this phase, right? It's crawling through the tissue, so it has this kind of funny spread out shape because it's moving, right? On this case, this is on the surface of maybe some plastic or something in the lab, so it's actually spreading out and moving around, looking for stuff to take in. Right? Then, once the dendritic cell has done that, and it's drained into a lymphatic vessel, right, now it becomes like the delivery truck. And the delivery truck is going to be moving now through the lymph vessel on its way to a lymph node. So morphologically, you can see the shape of the dendritic cell changes quite a bit. Right now, it has this kind of ruffled appearance to it. More interestingly is if you look at the staining, that red and green staining inside, what do you see? You still see a lot of yellow, right? So that's where MHC proteins are at the same location as lysosomal proteins. So that's where they're being loaded up by loaded up with small peptides, right? You see a lot more red. So this is sites where there's lysosomes now that no longer have the MHCs around them. And you see a lot of green, right? So this is where a lot of the MHC proteins have now spread away. And you say, well, where are they going? Well, they're going now towards the membrane of the cell, right? They've been loaded up and now they're going to the surface of the cell, getting ready to present stuff. Once they arrive, once the dendritic cells arrive in the lymph node, right, that is like the truck arriving at the mall. Now it's delivered the product. Now it's up to the dendritic cells to show it off, right, to any shoppers, so lymphocytes that are coming through the lymph node to browse and potentially buy, right? So look at the staining now, right? And you say, well, that's weird. What happened? How come the green is like smeared all over the place and now it's all like in these little protrusions? That's because at this point, all of the MHC has gone to the inner surface of the cell membrane, or not inner surface, but it's gone to the cell membrane, right? And is on the surface of the cell membrane now. And the dendritic cell has changed its shape dramatically to have lots and lots of spread out fingers to dramatically increase the amount of surface area it has on its cell membrane, where all of these MHC platforms can now be displayed, right? So that's why in cross section, it looks like this weird kind of smeary pattern. And here you see it. Here's the dendritic cell. It's beginning to really spread out. And these are T cells that have now synapsed with it and are checking out what the red dot is that the dendritic cells showing on its MHC, right? So in this role, the dendritic cell becomes like the salesperson, right? The salesperson who has to present the product in order for the buyer to have a chance to see it, get interested in it, and maybe actually make the purchase, right? So let's consider then what the T cell has been doing up until this point. Remember, the T cells, they're going between your blood and your lymph, right? So let us imagine that here we see part of a lymph node. So some T cells, some maybe we'll just call them like blue and green T cells, right, for lack of a better term. They've been riding through the blood. Maybe a blood vessel comes up behind the back of this cartoon, and now the capillaries of this blood vessel are flowing through the lymph node. Importantly, remember, this cross-section of a capillary here, this does not mean that at the end of this, it just suddenly ends and all of these green and blue cells are spilling out into the tissue. No, right? In reality, this is just a cross-section. So the vessel that we're looking at comes from behind your computer screen, comes straight at you through the lymph node, and then continues out, right? It's just that at these specific spots, at the high endothelial venule spots in the capillaries, right? These are the places where T cells can get the right kinds of signals to stop flowing through the blood, stick to the walls, and squeeze their way out, right? And the rest of the blood that was inside of this continues on. So in this particular case, let's see what happens, right? So once these T cells come into the lymph node, they are like the shoppers who have wandered into this store. They want to browse and see what's available at this store. So you have these dendritic cells that are now like the salespeople, and they're presenting things. So maybe in this case, this green T cell up here comes and checks out what this dendritic cell is showing it. But, you know, it's not something that this green T cell receptor is the right shape to get excited about, right? So as a result, what happens to that green T cell? It just moves on, you know, it bounces around, maybe talks to some more dendritic cells. It doesn't see anything. And eventually it wanders out of the store via the efferent lymphatic vessel, right? And now it's part of the lymph system, right? So now it hasn't gone back to the blood yet. It's still in the lymph system now, and it'll visit more and more lymph nodes, just like somebody wandering through the mall, going to a few different shops, right? Until eventually, if it doesn't see anything it wants, then it ends up being reintroduced into the blood up at the level of your shoulders, and then this process starts again. But let's look what happens to this blue T cell. This blue T cell that has a different T cell receptor than the green one, ooh, its T cell receptor is the right shape, right, to recognize the red dot that came from the site of infection. And this red dot's being presented on MHC2. So this blue T cell activates, right? It goes ahead, it hangs out, and it starts dividing to make many copies of itself. Right? And this T cell then will then begin to carry out its T cell functions. So if we back out a bit, right? So instead of being zoomed in on what's happening just at the lymph node, let's take a bigger picture. Right? So that means these blue and green T cells, they're doing their job by going between the blood and the lymph, the blood and the lymph. And so if you, I like this cartoon because it reminds you that if you were to follow one of these T cells going through the blood, right, at this particular part, the blood vessel forks, right? So it could go either way. So some of these blue T cells went this way, and this led them to come into the high endothelial venule that passes through this lymph node, and they exit, right? The other green ones here, maybe, and some of the blue ones went this way, right? They came to this lymph node first, but they didn't see anything in there that they liked. Even though the cartoon doesn't show any dendritic cells in there, there'd be some dendritic cells in here also. But the point is, right, there's nothing that interests these T cells because their T cell receptors are not the right shape to pick up on anything in here. So then they would leave this lymph node and be part of the lymph system now. Remember, the only way of leaving the lymph node is via the efferent or exiting lymph vessel that leads out of this one. And that efferent lymph vessel becomes the afferent lymph vessel leading into the next lymph node upstream, right? So why is this lymph node special? Oh, because this is the one that is closest to an infection, right? So the bacteria coming in, you have your dendritic cells that were at first kind of scrounging around in the tissue. They picked up the bacteria. Maybe they went ahead and broke it down into like different protein pieces, little peptide pieces, the red dots. Then these dendritic cells drained into this draining lymph vessel, right, along with all kinds of other debris and fluid, as well as maybe bacteria itself, right, travel through. That all arrives into this lymph node here. And so the T cells that enter this lymph node now have the chance to get excited because these blue T cells, right, have the right T cell receptor by chance to recognize the red dot. So these blue T cells, they are being activated, right? Whereas the green ones, whatever, right? They still don't see anything they like. They just pass on. Right? So those blue T cells that we were imagining as the shoppers who wandered into this particular shop at the mall and got excited about something that they saw in there, right? What is that process on the actual molecular level? What is that process of them getting excited about something that they see? And how do they really know, right? How do they actually get sort of, convinced to go ahead and activate against this thing? Right? So here we have an illustration of the T cell receptor. So this blue cell on the top is one of these blue T cells, right? So up until this point, it has never seen something that it gets excited about. But in this particular lymph node that was close to the site of infection, you have a dendritic cell, the yellow cell at the bottom, and it is presenting that red dot, that small piece of peptide that came from, let's say, the bacteria. And that small dot is the right shape for the T cell receptor of this particular T cell to get excited about. Right, so it binds there, the T cell receptor. Remember, it binds also to the MHC itself. So that's how the T cell knows that this red dot is actually being shown to it by another cell. And in this case, the dark purple thing is the CD4 coreceptor that then swings over and verifies that this is MHC2. Because this is a naive T cell, a naive CD4 positive helper T cell, right? That's waiting to activate. So its CD4 says, yes, we should activate against this because this is MHC2 that's showing it to us. Therefore, we know that this is some kind of a professional antigen-presenting cell. Because remember, a professional antigen-presenting cell that has MHC2 on it, that's actually only a small subset of cells in our body that do this. Only certain types of immune cells express MHC2. Nearly all of our cells can express MHC1, right? Because nearly all of our cells have the ability to raise the alarm that they are infected, right? And so that they might need to be destroyed, but only professional APCs like dendritic cells or macrophages or B cells express MHC2 because that's their job, right? Their job is to pass on a message that they found something, not that they themselves are infected by it, right? But our immune system wants to be careful because you don't want just random T cells activating against something that in reality is not really worth waking your immune system up against, right? So what if this red dot was in there, not because of an infection, but just randomly there's something left over? You don't need to get your immune system all excited about that. So that's why a naive T cell, like a CD4 T cell, specifically like this blue one here, needs an extra signal. And that extra signal is this B7 protein that's expressed on the surface of the dendritic cell. The B7 protein is like a salesperson badge that lets you know that this person is trustworthy, meaning when this person starts presenting something to you, right, that you can actually rely that this is a legitimate opportunity to go ahead and buy something. Let's say you go to Ala Moana because you want to, get a new iPhone or something like that, right? So you park in the garage, and as you're walking out of the garage, there's this kind of sketchy guy who's standing there with a big trench coat, right? And you walk by and he's like, hey, you want to buy a phone, right? And he has this brand new iPhone. What are they up to now? Up till 12 or something like that, right? And you're like, yeah, I did come here for a phone, right? An iPhone 12. And he's like, well, here you go, $200, right? You're probably going to be kind of sketched out, right? Because you're like, no thanks, I'm not interested, right? And he's like, all right, fine. But then where do you go? I mean, you go to the Apple store, right? You go to the Apple store because that's where the iPhones are, right? And in the Apple store, when you start talking to a salesperson, then you feel much more comfortable, right? Talking about the product, talking about the iPhone, and maybe even purchasing it. Right? So that's why it's very important. The dendritic cells during an infection, just like macrophages during an infection and B cells during an infection, in order for them to be respected as an appropriate antigen-presenting cell, right, they will start expressing this B7 protein on their surface. And that lets lymphocytes know that, hey, if I show you something here like this red dot, and this is the peptide that your T cell receptor, was designed to recognize, you can trust me because I'm showing you B7. So you know that this is a professional antigen presenting cell. And importantly, our body is currently experiencing an infection, right? So you need to pay attention to this. And the naive T cell has this receptor on its surface, CD28, that is the right shape to pick up the B7, right? So therefore, this naive T cell that up until this moment has never bound anything before, it wandered into this lymph node, encountered this dendritic cell. Its T cell receptor matches up with it. So the T cell receptor, along with the CD4, that represents one strong signal to get it excited. Right? But the other required signal is this B7C28, CD28. Therefore, this naive T cell knows this is no mistake, right? This is something I really have to pay attention to. How do these receptors then begin to signal? Right? So here we see the same illustration again, but kind of flipped upside down, right? So now the dendritic cell is on the top, showing the red dot on MHC class 2. And then at the bottom is the naive T cell with its T cell receptor complex. Here's the CD4. So the way this works is, remember I taught you that the T cell receptor alpha and beta chains themselves have pretty wimpy cytoplasmic domains, right? you can see they don't extend far into the cell at all. So that's not a lot of three-dimensional surface to really effectively interact with other proteins to kick off intracellular signals. Right? So that's why we have these other sort of helper proteins in the TCR complex. So on either side, we have these CD3 proteins, and then we also have this zeta chain protein. These proteins have substantially more cytoplasmic domain in them. And importantly, they have these amino acid sequences that are represented in yellow here. These different yellow stripes are what we call ITAMs, right? So ITAM stands for amino receptor tyrosine-based activation motifs. When this TCR binds to MHC class 2, what will happen? Right, so these two will come together and CD4 will have a chance to be brought close to it because the CD4 wants to bind to and become stabilized against the MHC2. This green protein that's on the cytoplasmic side of the CD4 coreceptor, LCK, is a kinase, right? So a kinase protein is a protein that will take phosphate groups and stick them onto other targets. In this case, this LCK would love to phosphorylate other proteins, right? It would love to put phosphate groups on other proteins. But if there are no other proteins close to it, it's not going to do anything, right? So that's in the first slide here, right? The LCK is just hanging onto the tail of the CD4, but there's no other proteins around it. So whatever, it's not doing anything. But if the CD4 binds to MHC class 2, then this LCK is brought up really close to the tails of the T cell receptor domain. And it will go ahead and start putting phosphate groups onto these ITAM regions. Now, I know it's terrible that the cartoon decided to make these red dots also, right? Because of all the things to do, they could have chosen any other color, but they chose the same color as the antigen up here, right? It's not the same, right? These are representing phosphate groups being added to these proteins. And what happens is that now the shape of these proteins are a little different. Right? This yellow protein that's been floating around somewhere else is called ZAP70. So ZAP70 has part of it that you can see here is just the right shape to bind to the cytoplasmic region of this zeta chain if the zeta chain has phosphate groups on it. Right, so as a result, the ZAP70 normally would just float by and disappear somewhere, but now it's the right shape to come and actually hang out for a little while because it binds to these phosphorylated ITAM motifs. Because of that, that means it is also brought close to the LCK, the green protein, and so the LCK is like, hey, I'm not done yet. You want some of this, right? And so he starts phosphorylating the ZAP70. And then once the ZAP70 is phosphorylated, it changes shape and activates and now starts communicating with a whole series of other proteins in the cytoplasm that ultimately lead to genes being turned on in the nucleus of this T cell and the T cell becoming activated. So this is a flow chart of those different pathways that the ZAP70, once it's phosphorylated, sort of stimulates. Right? I don't need you to memorize this, and in fact, I don't even want you to like get a headache trying to like spend too much time on this. But I want you to know two critical parts of it, because this part up here, right? This is where you have the naive T cell that's recognizing the specific peptide that's presented to its T cell receptor, and as a result, the T cell receptor complex has parts of it phosphorylated, right? And then the ZAP70 protein that was floating by binds to it and it gets phosphorylated. And now that phosphorylated ZAP70, you can see here, initiates a series of reactions. So now you can kind of just imagine everything in between is numerous different proteins talking to other proteins in a whole series of events. Eventually, this leads to certain transcription factors being activated. Transcription factors like AP1, which is a very common transcription factor that turns on many genes in our cells, and NF-kappa B. Remember, NF-kappa B was one of the first immune-related transcription factors that we looked at in this class. NF-kappa B turns on a whole bunch of genes related to immune responses, including things like cytokine genes. So this leads to the naive T cell no longer being naive, right? It is now woken up because it's been chosen. And so it wants to make copies of itself. So it begins to divide. Again, why? Because, well, its particular T cell receptor in the context of our whole body is very rare, right? Because this T cell might be the only T cell in our whole body that through VDJ recombination came up with this particular T cell receptor shape. And now it's been chosen as being valuable for recognizing this red dot that the dendritic cells are showing it. So you don't want to lose this T cell receptor because quite frankly, that would really suck, right? If after all of this, the T cell gets chosen and then it just died accidentally or something like that, right? So one of the first things that it does is it makes many copies of itself so that you have many copies of this T cell with the T cell receptor. And then it will differentiate. What does that mean? That means it finishes maturing into the particular type of, or get the certain types of abilities that it needs to finally carry out its function now as an activated helper T cell or an activated cytotoxic T cell. So this activation of CD4 helper T cells is very conservative. Hopefully you've gotten that impression, right? That whole point of needing to have a professional antigen presenting cell wear a badge, like a salesperson badge, the B7 protein, that lets the T cell actually see that this is a reliable message or a reliable signal that it's being shown, right? That's very conservative. So that's illustrated here. So normally on the left panel, you have the professional antigen presenting cell that maybe collected antigen from the site of infection. So this dendritic cell packaged it up, transported it into the lymph node. So this dendritic cell went through many changes along the way, from packaging things, delivering it like a truck, right? And now being a salesperson in the lymph node, and it's presenting this red dot. And so here is the naive T cell. It just so happens, boy, wow, this is a lucky day for this T cell. It came into the lymph node, browsing through, and sure enough, it sees what it likes, right? The red dot is what its T cell receptor is the right shape to detect, but right, it needs to trust that the cell that's showing it, right, is legitimate, right? And in this case, when we say that, it means this antigen presenting cell has to also have on its surface the B7 co-stimulator protein that's received by the CD28 of the naive T cell, and that combination of two signals stimulates the T cell to activate. All right, so that's what we've been talking about up until now. But what would happen if, for example, that same T cell wandered around and it encountered another cell that was presenting antigen, the same red dot on MHC2, right? But that other cell doesn't have the B7 co-stimulator protein on its surface. So there's nothing for the CD28 over here to bind to. In this case, it would get the signal from its T cell receptor that, hey, this red dot, this is what my T cell receptor is shaped to recognize, but it's not trusting what it's seeing here, right? Because it doesn't really know, wait, if we were really having an infection going on and this red dot does come from the foreign pathogen, how come this presenting cell, right, is not actually encouraging me in the form of wearing the B7? So without the B7, this T cell becomes anergic. Remember, anergic means it just shuts down and you lose this T cell, right? And you might say, well, okay, but that's kind of sad because couldn't you just wake this T cell up again if it turns out that this cell just needs to make B7 and it didn't get around to it or something like that? No, you cannot, right? Because even if this anergic T cell in the right panel here does in fact encounter another professional antigen presenting cell that is presenting the red dot and is wearing the B7 salesperson badge, you cannot wake up this anergic T cell, right? And you say, well, that sounds like a real waste. But this is just, again, another example of how our immune system, right, evolved to be very conservative about waking up things like T cells. Why? Because if you're sloppy about waking up T cells that shouldn't be woken up, and those T cells start to like talk to other immune cells and start mounting some sort of big SWAT team defense, you can imagine how bad that would be if in reality you really didn't have an infection to worry about in the first place. Or worse, right? What if this is a case of mistaken identity and this red dot actually is just some sort of normal protein, right? That this T cell improperly is getting excited over, right? That's why such a T cell that encounters MHC2 with a red dot without B7, showing it that it's a respectable salesperson, APC talking to it, your system just says, you know what, forget it. This T cell, let's just take it out of the picture and not worry about it. Right? All right. So when I talked about a helper T cell that does get properly activated, right, that does see the red dot that fits its T cell receptor and does see the B7 salesperson badge of the professional antigen presenting cell, because there actually is a legitimate threat going on, right? And the professional antigen presenting cell is saying, hey, we have an infection going on, so I need you to pay attention here, right? That T cell that activates the CD4 helper T cell now has to further differentiate and become a particular subtype or class of CD4 helper T cell, specifically suited to address what type of problem your body has detected. And when I say what kind of problem, I mean, well, obviously there's an infection going on, but what type of infection, right? Because, if you think about it intuitively, the types of ways your body will fight against a virus is very different than how your body might fight against bacteria or how your body might fight against a parasitic worm, right? So in order to coordinate the right type of response of all the different immune cells, you have to have the right type of CD4 helper cell. Right? So there are 5 classes of CD4 helper T cells that an activated CD4 T cell can become, depending upon the circumstances. So these are called Th1, Th17, Th2, TFH, and those regulatory T cells that we talked about last time, right? Remember, the regulatory T cells, their job is to go out, bind to cells that are presenting this target, but actually discourage other T cells from getting too excited about this, just in case this is just sort of like a false, noisy kind of signal, right? But let's look at these other 4 types here. For now, I don't want you to worry about this middle part, because this talks about the types of cytokines that these T cells are receiving in order to become this particular subtype of T cell. For now, I just want you to focus on what these different classes do. Right? So Th1 cells, their job will be to go out and talk to macrophages and stimulate macrophages to increase their activity. Th17 cells talk with neutrophils, right, to help coordinate a stronger neutrophil response. Th2 cells, they activate and work with our antibody response from our B cells in order to respond to things like parasites. And TFH cells, the FH stands for follicular helper cells, they hang out. They hang out in the lymph nodes to help B cells that are coming in get properly activated, right? And so in that sense, they also help with a much stronger antibody response. So those are activated CD4 cells, and we'll look at this later next time. But what about the CD8 cells, right? CD8 cells oftentimes require even stronger signals to convince them to activate. Why? Well, it's one thing to activate a CD4 helper T cell improperly, right? Because then that cell, well, it just starts talking to other immune cells that aren't really necessary for it to talk to, whatever. If there's nothing really that comes out of it, then maybe that signal just goes away. But if you improperly activate a cytotoxic CD8 T cell and it goes off and starts killing cells in your body, right, that are presenting whatever signal it was that it improperly got excited about, that could cause a big problem, right? Because these CD8 cells, they just synapse with another cell. If it sees what it's looking for, it tells that cell to die. Then that CD8 cell just moves on to the next cell, kills that one, moves on to the next one, kills that one, right? Normally, right, that's what you want if these cells are infected. Right? So in this case, a naive CD8 T cell that binds to something like this is a dendritic cell, right? This dendritic cell is presenting MHC1 on it because it has the red dot as a function of it being infected. Right? This CD8 cell, even though it also sees the B7 salesperson badge, that's not enough to convince this CD8 cell to be activated. It has to be that nearby there's a helper CD4 T cell that's seeing the red dot being presented on MHC2, right? And that will convince this helper T cell to release interleukin 2 or IL-2. And that's what's needed to go ahead and activate the CD8 cells, right? So a much more conservative approach to making sure that these killer T cells really are properly recognizing something that should be of concern. On the left are some other, some instances where, depending on what kind of virus is actually infecting the cells, just detecting the virus infected cell is enough to activate the CD8 cell. So certain types of viruses that we've sort of been with us through evolutionary history, right, and time again have shown that if CD8 cells activate against these dendritic cells that are infected, that's fine, because that's the best way to go ahead and not waste time and just go ahead and start looking for other infected cells, right? So certain types of virus peptides will activate CD8 cells by themselves. Okay. All right, so that's it for today.